Hello my dear students, welcome back. In this session, I'll be discussing about Tryptophan Operon. The last session we completed lac operon, which is an inducible operon. We already discussed what is an inducible operon, where there is a substance inducer is present. In presence of inducer, the transcription will take place. That is called inducible operon. And inducible operons are generally, they operate during catabolic process. The operons which operate during catabolic process, they are called as inducible operons. Like for example, lac operon. Now here, tryptophan operon is called as a repressible operon. Repressible operons generally they operate during anabolic process. That means synthetic process. So this operon is for synthesis of tryptophan. So it is called as a repressible operon. So here if tryptophan, which is the end product, is present, or if it accumulates in large amount, or if it is added to the medium, then the transcription will be switched off. So in presence of tryptophan, transcription will be switched off if it is in excess amount. This type of operon where end product will stop the process of transcription, or it will regulate the process of transcription, that is called as a repressible operon. Now let us see what are all the composition of tryptophan operon. The first thing, tryptophan operon consists of structural genes. There are 5 structural genes are present. They are named as E, D, C, B, A in reverse. They are named. So these are the 5 structural genes. These 5 structural genes will produce the enzymes which are required for synthesis of tryptophan. So these 5 structural genes together, they form a single mRNA. So mRNA has the information for synthesis of different enzymes which help in synthesis of tryptophan. So this is called as a polycistronic gene. It is called as a polycistronic gene which will synthesize a polycistronic mRNA. So first I give you the composition, then the structure and how it functions. After structural genes, next it is called as an operator gene. After structural genes, it is called as operator gene. Operator gene is present towards, upstream to structural genes. It is present upstream to structural genes. Upstream position already you know, it is towards the 5' end of the coding strand. It is present upstream to structural gene. And this operator gene always will be in functional state unless the end product, that is tryptophan, accumulates, which will block the operator. How it blocks, we are going to see. So when tryptophan is not present or present in low quantity, this operator will not combine with repressor. You know, repressor and operator, when they combine together, then transcription cannot take place. But when operator is left free, then transcription can take place. So it is always left free. It is always free unless tryptophan increases in the medium. That is the operator gene. Now next is called as, next component after operator, it is called as a regulator gene. It is called as a regulator gene. Now regulator gene actually is not present along with these. it is present separately. And this regulator gene represented as tryptophan R. This is regulator gene. This regulator gene is a constitutive gene. It is a constitutive gene. That means it is always functional gene. It is a constitutive gene. This regulatory gene, it produces aporepressor. This is a protein called aporepressor, which is inactive form of repressor. Aporepressor is an inactive form of repressor. This aporepressor must combine with co-repressor. Aporepressor must combine with co-repressor. So what is co-repressor here? It is the tryptophan, that is the end product. Tryptophan or the end product is called as a co-repressor. When aporepressor combines with co-repressor, then it changes to active form of repressor. It changes to active form of repressor. When it changes to active form of repressor, then this active form of repressor can bind to operator and then transcription cannot take place. So this association of aporepressor, co-repressor is very, very important here. That is called as it forms an active repressor. So this formation of active repressor takes place when there is an end product present, that is tryptophan is present. Now the next gene here, next gene is called as operator gene. Operator we just completed, promoter gene. Next one is called as a promoter gene. This promoter gene, or P, we can represent it as a P, promoter gene. This is also present upstream to structural genes. Upstream to structural genes. This is also present upstream to structural genes. That is called as a promoter gene. Now this promoter gene is very important for RNA polymerase to bind. What is the function of promoter gene? Here RNA polymerase enzyme which helps in transcription, it binds to promoter and moves towards structural genes, through operator, operator. That is called as that is the function of promoter. So if operator is left free, RNA polymerase easily can move from promoter to the structural genes and help in the transcription of structural genes. That is the function of a promoter region. So now let us see the last part component, which is called as a co-repressor, which I already mentioned, co-repressor. Co-repressor is the end product. End product here is called as co-repressor. And this co-repressor, which is the end product, what is the end product here? It is the tryptophan. Tryptophan is the end product. That functions as co-repressor. So when co-repressor binds to aporepressor, it converts aporepressor to active form of repressor. That is the function of co-repressor. Okay, now let us see diagrammatically how the genes are switched on and off. First, let us see how the genes are switched on. That means how transcription will take place. You know, if transcription has to take place, then the operator has to be free. So let us see first diagram where operon will be on. first diagram where operon will be on. On means transcription will take place and it is going to produce the protein. So this is the first, second, third, fourth, fifth gene. And this is the operator and this is the promoter. So let me write it here. So this is A, this is B, C, D and E. This is the operator and this is the promoter. And here there is a regulator gene. Now this regulator gene, this regulator gene will produce aporepressor. And we know aporepressor, it is the inactive repressor. Aporepressor will be the inactive repressor. So here when it is inactive repressor, now what will happen? This operator will be left free. So now transcription can take place. So these genes will produce mRNA. These genes will produce mRNA. That means this is the gene E and D. Say this is C, B and A. So these genes will produce single mRNA in this direction. Single mRNA. Now this mRNA is going to produce different enzymes. This mRNA will produce different enzymes for synthesis. So here we can see the substrate here is substrate for synthesis of tryptophan is called as choristmic acid. Substrate for synthesis is called as a choristmic acid. So this choristmic acid, first it will produce say 5 types of enzymes. So this is how it will produce 5 enzymes. And these enzymes, so first 2, this is represented by, say these 2 genes will first produce enzymes here. This C will directly produce this, and again these 2 we take. Again these 2 we take if they are going to produce this way. So in this way 5 different enzymes will be produced. Finally, to synthesize the amino acid tryptophan. This is this is by this process we can name it. This is transcription, and this process is translation. This process is translation. So finally from choristmic acid to tryptophan is formed. This is when operator is switched on. So when will be the operator switched on and transcription can take place? When operator is free. This is very important. Operator is free. Operator will be free when repressor cannot bind to operator. So repressor cannot bind because in the form of aporepressor, which is the inactive form. I hope this is clear, children. You have to just copy it or look. Then the same diagram I will change it to when operon is switched off. Let us see how do we draw the diagram. When operon is switched off. When it is switched off, first thing we can say there is no transcription and no translation taking place. There is no transcription and translation taking place. So what happens here? The aporepressor will combine with co-repressor. Aporepressor will combine with co-repressor and then it will change to active repressor. When it changes to active repressor, this active repressor will bind to the operator. So when active repressor binds to operator, there is no transcription, that means genes are switched off, and therefore no translation also. There is no transcription and also there is no translation. So the gene is said to be, it is switched off. So this is the aporepressor co-repressor system in case of tryptophan operon. So now briefly how we can write is, say when the presence of inducer, what will happen? Absence of inducer, what will happen? Or if co-repressor is present. If co-repressor is present, what is it getting converted into? If co-repressor, if we write. So we can write it in this way. If co-repressor is present, which is the end product, the active repressor can be made inactive. Suppose, let us write this way. Suppose there is an active repressor. To summarize, this is how we can write. Suppose there is an active repressor. This active repressor can be converted into inactive repressor. So when will it be in inactive form? When there is a transcription taking place. That means when there is inducer present. What is the inducer here? Inducer is the substrate. We can say corysmic acid. Corysmic acid should be the inducer. So if substrate is present in large amount, then we can say active repressor is converted into inactive repressor. The same thing, suppose if we take how inactive repressor converted into active form. If we take inactive repressor is present, inactive repressor. What is inactive repressor? It is aporepressor. It can be converted into active form, active repressor. Inactive can be converted into active repressor in presence of end product, that is in presence of co-repressor. In presence of co-repressor. Inactive repressor can be converted into active in presence of co-repressor. Co-repressor here is the tryptophan. So that is how we can, to summarize, we can say the conversion of active inactive repressor. So with this I conclude the tryptophan operon, my dear students. So here I will conclude the topic. So next session I will take some other topic from this chapter. Mostly I will be continuing with DNA. In my very first video I just discussed briefly the DNA structure. So I think I will continue with DNA structure and replication in my next video. So completely about DNA we are going to discuss in some 4 to 5 videos. So thank you very much children. Those who have still joined my session for the first time, please watch it very carefully from the beginning to the end, and after understanding then you subscribe my channel and share to your friends. So thank you very much. Have a good day.